Our next speaker again is Matt Corniss. And thanks a lot, Matt, for being able to cover. I know, unfortunately, Dr. Brian Roth has some health issues and wasn't able to cover the Huron, Michigan diet study info. So we got Matt pinch hitting and anxious to see what you have to say about the diets.
Yeah, thanks, Dan. I know Brian sends his regrets about not being able to make it. I did stick a photo of him here on the front so y'all can see what he looks like. He does like giving these talks to these groups, and unfortunately, he just his health right now would not enable him to do so. I've been a partner on this diet project. The first inception was actually in 2015 and 16, led by University of Milwaukee and Purdue, and then the Roth Lab kind of took over diet work starting in 2017, and we've been partners since. Now, so I have a decent sense of what Brian's results have been. I've tried to study this slideshow this afternoon, making some tweaks here and there, but forgive me if there's some places where I stumble, as I haven't given this talk ever. But I do want to start by recognizing we have lots of collaborators going into this. Mackenzie Smith in particular and Sarah Ang are some of the key players in Roth's lab, as well as kind of an army of undergraduates that are doing the diet work. Lots of help from all of the state DNRs, Fish and Wildlife Service, Wisconsin Sea Grant, volunteer anglers and sport fishing groups, and then the trawl crews from USGS and MDNR. Really, it's become kind of a diet clearinghouse for Lakes Michigan and Huron. So the Great Lakes have changed a lot over the last several decades via habitat degradation, overfishing, and invasive species. We have some examples here where we see kind of this is the yield of lake trout. And we can see that just kind of crashed around 1950 as a result, in part of fishing and in part because of invasive sea lamprey, which we see here in this photo. Given all of that change, there's been a lot of uncertainty in diets. One of the management goals is to improve the predator-prey ratio model, which is used to help inform stocking decisions in Lake Michigan, to incorporate multiple species. That model historically has been comparing Chinook salmon to alewife, but as we start to see changes in the biomass of these species, I know we are starting to see more steelhead and more lake trout. There's been fewer Chinook salmon, so there's a need to look at multiple salmoning species in this model. This model also doesn't account for what predators are actually eating, and we're hoping to have a better accounting of predatory demand. Because of all this, we need to know the diets of predators. So looking at what these predators eat, obviously prey represents a key input in calculating predator demand. The current models are lake-wide, which seems very appropriate for specialists like Chinook salmon and coho. They are largely mobile species feeding on alewife, but we're not sure how well that model works for generalist feeders like lake trout and steelhead, where we've seen more adaptable diets. So if we look at past work that's been published about what predators eat, we do see that Chinook salmon, coho salmon, and steelhead are more homogeneous. That means their diets are very consistent. They focus on alewife, whereas brown trout and lake trout are more generalist. They eat alewife, but also some of these bottom-oriented species like sculpin and round goby. All the species do prefer alewife, but when we see low abundance of alewife is when we start seeing interesting patterns emerge for brown trout, lake trout, as well as steelhead. So there's some implications here for the sustainability of the fisheries, stocking in terms of species composition and quantity. And so again, this comes down to understanding diets. So some of the recent history here, there was a comprehensive diet study in Lake Huron from 2009 to 2011 by Roseman et al. There might have been some data in there from Lake Michigan as well. The 2015 and 16 studies, when I started to get involved, that was on Lake Michigan only. And then both lakes, we started looking at that in 2017. The study is ongoing, but Brian didn't have time to update the presentation with data from 2023. For currently, this presentation goes through 2021. In order to really get a handle on some of the variability in diets, it requires sampling not only across species, but across different regions of these lakes and seasons. So the questions that we're trying to address today with Brian's talk are: what do predatory fishes eat in Lakes Michigan and Huron? How do foraging conditions differ across the two lakes, and have either changed through time or across space? This is where partners are really critical in terms of collecting the stomachs for these analyses. Anglers that are sampled via creel clerks at tournaments by Fish and Wildlife Techs, or just individual donations, as well as agency and tribal gillnet surveys, have really produced a lot of stomachs. In terms of our sample sizes, again, there was restricted collections in 2020, and then as Brian said, they're still entering and analyzing 2023 data. Special shout out here, the Fish and Wildlife Mass Marking Program provides a lot of stomachs, as does the Michigan Department of Natural Resources. But if you look at everything combined on Lake Huron, the total sample size dating back to 2009 is diets on about 13,350 fish. And in Lake Michigan, it's about 12,000 fish dating back to 2015. I mentioned spatial sampling. So to look at breaking down these lakes into regions, Lake Michigan has kind of been split into the four cardinal directions: northwest, or NW, southwest, or SW, northeast, or NE, southeast, or SE. In Lake Huron, we only looked at the US waters, and that's been split up into northern, which is NH, Saginaw Bay, and southern, which is SH. So in order to analyze these diets, Brian's lab is visually identifying the contents in the stomachs, identifying prey fish to species, invertebrates to order, and then measuring and weighing the contents. Prey length can be back calculated using the vertebrae or clythra, which are different types of bones coming off of these fish. Those are also helped used to identify fish. And then weighing each taxa, the diet proportions are going to be expressed as a wet weight. And then fullness is expressed based on the mass at capture. So identifying stomach contents is not always an easy task. They're often very digested. This Petri dish you can see here is blown up in size with a yellow arrow, and it just kind of looks like a bunch of goo. So in terms of how we can tell fish species apart, a lot of times we're looking at the clythra. Those are the pectoral fin supports, the otoliths or ear bones, or the caudal bones. So we see some pictures here. These top pictures here, I believe, are clythra. We see otoliths here in the middle. This picture here on the left might be a caudal bone. Might not be. I'm not someone who identifies species from bones, but I know that it's something that lots of people are very well trained to do, especially in the Roth lab. So in terms of our results, part one is: what do predatory fishes eat in Lake Michigan and Huron? Brian started this off with a couple of fun pictures. On the left here, we have what appears to me to be rocks or perhaps charcoal. There's some sort of bird foot here in the middle, Coca-Cola can here on the right. And then I had to add one from a few years ago up in Lake Superior. There was a lake trout caught with a bratwurst in the stomach. So these are obviously exceptions, but you never know what you're going to find when you're looking inside a fish stomach. In terms of summarizing these diets, you're going to see various abbreviations. ALE for alewife, that's going to be light blue. RGB for ongoby, which will be dark blue. Invertebrates will be light green. Smelt will be dark green. Corriganines will be pink. Sculpins in red. Other, which is kind of a catch-all, will be purple, and that's basically other fish or unidentifiable goo. And then YEP would be yellow perch, and that's going to be in a light blue as well. Actually, I think that's going to be in yellow. I think that color is wrong here. Yeah, it's on yellow here in these figures. So getting into the results here. So what you're seeing here are the diet composition of fish in Lake Michigan. From top to bottom, we see brown trout, Chinook salmon, coho salmon, lake trout, and steelhead by year. These diets are the proportionate diet by wet mass or wet weight. And some general patterns: if you look at that lake trout bar, you see a lot of dark blue. Those dark blues are round gobies, and we see lake trout here are consuming more round gobies than any other predator in Lake Michigan. The light blue, of course, is alewife. We see everybody eating alewife, but the lake trout diets are a little more diverse. If you look at steelhead, especially in 2015 and 16, we saw a consumption of a lot of bugs. Those are the light greens there. Chinook and coho diets were fairly boring, pretty much alewife, with rare exception. I think this really interesting one for coho in 2023 is probably based on low sample size for that year at the time that this figure was made. If we look at Lake Huron, things get a lot more interesting. So these are the diet data from Lake Huron, again by wet weight. And so you see a lot more color. That indicates a lot more diet variability. Somehow, if you look at Chinook salmon, we do still see them finding alewife. Those are the light blue, but pretty much everything else in Lake Huron is not really relying on alewife anymore, and that's because alewife are at such low numbers in Lake Huron. If we look at Atlantic salmon and brown trout, the sample sizes were pretty low. Those are the two species at the top. So, take that with a grain of salt when you're looking at these data. Looking at Atlantic salmon, we see a lot of round gobies and smelt, some invertebrates. This orange bar, that's a catch-all of just fish. Brown trout, we're seeing gobies and invertebrates as well. We see more smelt. Smelt are more important in all these diets. That's that very pretty emerald green that you're seeing in most of these. Round gobies are still prevalent. In fact, they're more prevalent in the diets in Lake Huron. That's still that dark blue. We see a lot of those in lake trout as well as walleye, brown trout, Atlantic salmon, and we do see a few popping up in steelhead and coho as well. And then here, the steelhead really like bugs. You can see that light green for steelhead is the vast majority of their diets in most years. And one thing I want to note here, I think I added this here. So this is actually something I added when I was going through the talk. This is a data figure from Ben Tershak et al. What you're looking at here is steelhead diets in Lake Michigan. And what I want to do by way of example is if you look at the gray lines here. In 2014, that's gray, we see very little small alewife. And we see very little large alewife in the diets up until a certain size. And as a result of that, we see a lot of bugs in the diet. By contrast, in 2016, that's blue. We don't see very many bugs. We had a lot more alewife in the lake, and that's what the steelhead were eating. So the reason I added this here is because this paper really is showing that steelhead, among other animals, their diets change based on how many alewife are abundant in a given year. And of course, in Lake Huron, we don't have a lot of alewife, so I think that's in part explaining why we see so many bugs in the steelhead diets. So some contrasts between lakes. At the top here, we have lake trout diets in Lake Huron versus Lake Michigan in the bottom. Much greater dependence on round goby in Lake Huron. Smelt instead of alewife are the dominant pelagic forage in Lake Huron. And then we actually do see some coregonines in pink here in Lake Huron, although they are a small component of the diet. If we look here at steelhead, again, pretty substantial differences between Lake Huron in the top and Lake Michigan in the bottom. That abundance of alewife in Lake Michigan really does appear to be a driving factor. We don't see a lot of alewife in the Lake Huron diets. It's mostly bugs, smelt, and again, coregonines in pink, with a smattering of other things in Lake Huron. Whereas in Lake Michigan, at least from 2017 through 2021, it was largely alewife. And then even Chinook salmon, we do see some diet variation. We know these are pelagic obligate feeders. That means they don't look down to eat. So in Lake Michigan, that's why they're pretty much eating universally alewife with a smattering of coregonines thrown in here in pink. In Lake Huron, they are still finding alewife, and they're supplementing that with smelt in dark green and invertebrates, which are mostly spiny water fleas, also known as Bithytryphes in light green. So how are Chinook salmon finding alewife in Lake Huron? If we look at kind of the spatial pattern here for Chinook salmon, one thing Brian noted here is almost all of the Chinook salmon were captured in northern Lake Huron. I happen to know that a number of these came from mass marking, and we tend to see most of those fish captured there during the spawning run. So it's possible that we're seeing fish that were feeding in en route and then pulling out alewife from their guts once they have arrived in MH1. Once we move to Saginaw Bay, which is the middle of all these figures, and then the southern Lake Huron, which is the right of all these figures, we do see the Chinook salmon diets changing a good bit. We also saw diets differing across space. So again, these middle bars are Saginaw Bay. If we look here for Atlantic salmon and Chinook salmon, we saw a lot of smelt in the diets here in Saginaw Bay, whereas northern and southern Lake Huron, which are the bars to the left and right of this bar circled in yellow, have different diet patterns. Likewise, for lake trout in northern and southern Lake Huron, they were eating a lot of round goby. Same for walleye, but somehow in Saginaw Bay, we saw a different pattern with walleye consuming a lot of yellow perch, and lake trout consuming a lot of, well, it just says fish. So presumably those are fish that are not round gobies or smelt or cannot be identified. So Saginaw Bay, as those who fish it know, is a different system in terms of the abundance of different prey species. So the next thing Brian's lab asked is, do we see regional diet diversity in Lake Michigan? So these figures are pooled across years. Largely, we just see alewife, but there are a few patterns worth highlighting here. For lake trout, which is LAT here on the right, round goby consumption was the highest in Northwest Lake Michigan and the lowest in Southwest Lake Michigan. And then there was this pattern with Eastern region brown trout having more diverse diets in the Western region. That could be related to sample size, but otherwise we're just seeing mostly alewife. This next figure here is looking at where are lake trout consuming gobies in Lake Michigan. And actually this is when, so these are seasonal effects. Each of these numbers correspond to the month. 4 is April, 5 is May, etc. And so what we see here is there is a decrease in round goby consumption through the summer. So you can see in all of these regions, those dark navy blue bars, we see them towards the left of the figure when it's springtime. That transitions into alewife in the summer. And this is largely because round gobies undergo a migration. They migrate out to deeper waters in the fall. They overwinter where it's deep, and then they migrate back in the spring. So during fall and spring and winter, they are in deeper waters where they're available as food. During the summer, they're in waters that are usually 5 to 10 meters or less, and they're spawning under rocks. This is a photo of a goby from a rock from a river where this goby was spawning in literally like 6 inches of water. So obviously that's not a spot you're going to find a lot of salmon and trout in. So we think that this migration pattern is playing heavily into the seasonal diet patterns for lake trout in Lake Michigan. In steelhead, we did see generally decreasing the contribution of invertebrates through time. Here, the x-axis is years. And again, if you take a close look at that published work by Ben Tershak, he did a really nice job of relaying these patterns in alewife and invertebrate consumption to the abundance of alewife in any given year. Steelhead diets by month here in Lake Michigan. We do see a consistent shift from bugs to alewife as we get closer to summer. So again, spring is towards the left, lots of bugs, more alewife in the summer. Part of that could have to do with the alewife migration pattern. So alewife come into shallow waters in June, early July, late May, right around then for spawning. And so by the time we get to kind of late June, July, August, September, those alewife are back offshore, likely where they're more available to these predators. All right, so the phase 2 of this study is looking at foraging conditions. I'm going to highlight patterns in the number of empty stomachs, which is a potential indicator for the difficulty in finding prey. Prey diversity, which is the total number of prey species found, and ration size, which I will define when we get there. So this is a figure showing the proportion of empty stomachs for brown trout, BNT, Chinook salmon on the upper right, coho in the central left, lake trout in the central right, and rainbow trout or steelhead in the bottom left. You can see for all of these over from 2015 to 2019, a general declining pattern in the amount of empty stomachs. And this is consistent with the predator-prey ratio data. So if I just kind of show you those data real quick, the predator-prey ratio is based on modeled estimates of Chinook salmon biomass, which you see in the top, alewife biomass in the middle, and the ratio itself in the bottom here. The yellow box here is the 2015 through 2019. So what we see here is in Lake Michigan anyway, a reduction in Chinook salmon biomass, an increase in alewife biomass, and then a decrease in the number of predators for available prey. So in other words, predator-prey ratio suggests there's more prey available per predator, and the empty stomach pattern is consistent with that. So in terms of diet diversity, this is characterized by species richness. That's just a count of the number of species in the diet. The Roth Lab has observed that this is increasing through time lake-wide for Lake Michigan, with some significant region and species effects. So if we kind of dive into this figure a little bit, generally there was increasing diet diversity over time, going from 2015 through 2021 for coho salmon, which is green, lake trout, which is blue, and steelhead, which is pink. We also saw diet diversity across species increasing in southeastern Lake Michigan. And there weren't very many region species relationships that were significant here for diet diversity in Lake Michigan. Looking at diet diversity in Lake Huron, they integrated historical data from the Roseman et al. Paper. Most species and region combinations were higher in more recent years, particularly if we focus on coho salmon, lake trout, rainbow trout, and walleye. We can see this increase in species richness. Diversity was also generally higher in northern Lake Huron than in Saginaw Bay or southern Lake Huron. Next, we looked at ration, and this is trying to address the question of, has the size of the meal changed? So ration is basically a calculation of the weight of prey in the stomach divided by the weight of the fish. And so if you average that out, you get a sense of just how much food is consumed in a given meal. Ration has been increasing in Lake Michigan for coho salmon and for steelhead. And those are highlighted here in blue and in pink. Those increases aren't huge, but they are present. We also have seen increasing ration size over time from 2015 through 2021 in, generally speaking, in northwestern and southwestern Lake Michigan. However, we did see ration decreasing for Chinook salmon, rainbow trout, and lake trout in northeastern Lake Michigan. Why we're seeing these patterns, I can't say, but these are patterns that have been detected in the diets. There were some significant regional trends. Those are highlighted by an asterisk here. So the ones that are actually statistically significant are increase and decrease for brown trout in northeast and northwest Michigan, respectively. And then patterns in southeast and southwest Michigan for coho salmon and steelhead. If we look in Lake Huron, we do see kind of mixed results in northern Lake Huron. Again, we're looking at ration here on the y-axis by year. In this case, because we have those historical data from the Roseman et al. Paper, we go all the way back to 2011. We see increasing trends for Atlantic salmon and rainbow trout and walleye, decreasing trends for coho salmon and Chinook salmon, and stable trends for brown trout and lake trout. Generally, though, in Saginaw Bay and southern Lake Huron, we saw decreasing trends in the size of the meal. And we did see that Chinook salmon in Lake Huron were doing poorly everywhere, with decrease in their ration size in Lake Huron in all three regions. So to summarize what we talked about today in terms of diet composition, we see alewife as the dominant prey item in Lake Michigan, with a lot more gobies and smelts in the diets in Lake Huron. Diets were relatively consistent when we looked at species patterns. They did vary regionally and seasonally in some cases. But, we do have this type now where we have, Chinook and coho really relying on alewife. Steelhead will rely on alewife when they're there. Otherwise, they'll eat, invertebrates. And then the lake trout and brown trout really focusing on gobies when alewife are not present. There was some regional variation again in both lakes. Some examples: Chinook salmon in Lake Huron, where they had alewife in northern Lake Huron in their diets, but mostly smelt and other items in the south. And then lake trout in Lake Michigan, where we had few gobies in the southwest relative to other regions. In terms of the findings on prey diversity, they were generally increasing through time in Lake Huron and increasing in southeastern Lake Michigan. If we look at the ration size or the size of the meal, if you characterize this across both lakes, it was generally stable or increasing, with some exceptions being a decreasing pattern in northeastern Lake Michigan over time, a decreasing pattern in southern Lake Huron over time, and Chinook salmon in Lake Huron in general decreasing in ration size over time. So in some of the implications that are highlighted, the prey conditions in general appear to be improving. Evidence is showing that the predators are consuming more and more diverse prey in general, again, with some of the exceptions noted on the last slide. We do see that regional differences are apparent. Species richness, ration, and composition can all differ by region, and this depends on species. So some conclusions here. We know these data are useful for modeling. One of the questions that we're asking is whether the lake-wide averages that are currently being used, whether those are adequate or not. We also know that goby consumption by lake trout is a source of uncertainty, particularly with the seasonal and spatial patterns there. Some of the remaining questions include how do diets relate to prey availability? And that's the subject of the most recent ongoing grant to the Roth Lab. And then how to best incorporate known diet variability into predatory demand models. In other words, how do we incorporate seasonal and spatial diet variability into these models? And perhaps the question of, does this variability matter in terms of what those models are telling us and informing managers in terms of their policy decisions? How can you help with this study? Big bold letters of please, please, pretty please with a sugar on top. Donate stomachs to this study. The more stomachs we have, the more data we have. This is especially valuable to us in the shoulder seasons, which would be spring and fall, or even winter if you're out there ice fishing. Most of the samples we get from the agency surveys are going to be from summer. We don't see a ton of brown trout anywhere, so anytime you catch a brown trout, if you can donate a stomach, that's going to be a relatively uncommon data point for us. And we, being the Roth Lab, are happy to provide supplies and any training necessary to help with this. If you have any questions, I will try to help answer those that I can in the next few minutes. Otherwise, this is Brian's email address and the Roth Lab website. I encourage you to reach out to him as well if you want to engage in a conversation direct.